Delighted to welcome Ciara. She's recently started her lecturing post within the school just at the beginning of the month, so we're delighted to have her present today. So thanks very much, Ciara.
Thanks, Amanda. So, thanks for having me, everyone. I'm delighted to be here. Okay, so I'm going to talk today about neural repetition effects during recognition memory. So we've known for quite a while that repetition of stimuli typically decreases neural response. So you present a stimulus once, you get a particular response. You present it again, typically you get a reduced response. And this is called repetition suppression, or RS. And there's been a number of theories as to what causes RS. It's been linked to more efficient neural processing, and it's also been linked with behavioural priming. So we know, for example, that when we're presented with a stimulus and then we'll see it again later on, we respond more quickly and more efficiently to it. So repetition suppression seems to be a kind of a neural reflection of that behavioural priming effect. There's some suggestion that neurally it might be due to a sort of a sharpening of neural response. So information, when it's presented the first time, is represented by a certain configuration of neurons, and when it's presented a second time, it's going to be represented by a less dense representation, so a sparser representation. So this is a figure here from the Henson and Rugg paper in 2003, who first really described in detail neural response suppression. So to take the example here of the stimulus of the face, so we see it the first time, and what we see normally in PET or fMRI studies, we see the image on the very right here, we get a bold signal, so we see an amount of signal, an amount of response. And we get a certain amount of response the first time we present the stimulus. And the second time we present the stimulus, we see a reduced response, or a reduced strength of the bold signal. And the third time, we see a reduced response yet again. And Henson and Rugg theorised that this has to do with this kind of sparse representations. They're showing this using this middle line of the neuron. So the first time the face is presented, we see this whole network of neurons responding. The second time, a few of those, sort of as more sparse representation, fewer of those neurons are required to represent the stimulus, and the next time fewer again. So the idea is that this is kind of an efficient method of the brain representing stimuli that it's seen before. So the first time it sees something, it's all hands on deck. A lot of representation is required, and for subsequent representations, we need fewer neurons. We have a more kind of, it's the equivalent of sort of a compression algorithm, like a JPEG for photographs. You need less information the second time around in order to represent the image. Okay, so in typical recognition memory paradigms, participants are presented with a series of stimuli, photographs for example, or any kind of images, and are instructed to explicitly identify the stimuli as being old or new. I've seen this before, I've never seen this before. And we know that presentation of old, familiar, previously viewed stimuli typically results in repetition suppression in the medial temporal lobe, and specifically in the hippocampus. But this isn't always the case. That's the typical finding. But there are other findings that actually show the opposite effect. So the representation of a previously viewed stimulus actually resulting in an enhancement of neuronal response. And we call this repetition enhancement, or RE. So what we're seeing here is effectively two neural codes of memory. It's the recognition of a stimulus you've seen before. It's typically represented by repetition suppression, RS, but sometimes represented by repetition enhancement, or RE. And really what my colleagues and I wanted to determine was, what is it that determines whether we see repetition suppression or repetition enhancement? So I'm going to present a series of experiments in which we try to sort of delve into that. Okay, so this is what we want to know. Why does repetition of stimuli sometimes result in an increase in neural response, but more usually in a decrease? Well, repetition enhancement might occur when the stimuli are unfamiliar. So it's been seen before, but it's relatively novel. Repetition suppression, it's been hypothesized, seems to exist in order to ensure that contextually novel stimuli can elicit an appropriate response. So what I mean by that is, when we see something new, there's a big response, as I showed you in the first figure there. Okay. So the idea is that repetition suppression, this more sparse representation, occurs in order to allow a more strong, contextually appropriate neural response to novel stimuli. So it's kind of repressing the old stimuli in favour of the new stimuli. So we hypothesised that RS and RE might actually reflect the nature of prior mnemonic experience with the information. So we're interested in how your previous experience, your learning experience with the information influences whether we see RS or RE. So in our first experiment, we decided to explicitly examine this. So we wanted to isolate the role of encoding experience on neural response to repetition. And because we know that typically we see very strong repetition enhancement in response, or at least we see enhancement of response to novel stimuli, we decided, well, we want to rule out novel stimuli altogether here. We just want to look at previously viewed stimuli, and we want to look at how differences in the encoding experience might influence the presence of RS or RE. So what we did was we compared the response to repetition of items that had previously been committed to memory, and we called them the WM or working memory set, with items that had previously been seen but not memorised, and we called those the primed set. And crucially, we used an incidental recognition paradigm, so we presented the stimuli in such a way that the participants were not explicitly instructed to identify the stimuli as old or new. So we weren't directing their attention to the novelty or familiarity of the stimuli in a top-down manner. We really wanted to see whether we could elicit a sort of bottom-up response, irrespective of intentional orientation to retrieve the information from memory. So we used stimuli like these. These are just, as you can see, black and white, abstract shapes. These were originally JPEGs of just common items, so a boat, a house, a car, a wheelbarrow, and so on. And using a particular algorithm, we just manipulated them to make these abstract shapes. So the idea here is that they're very individual, they're easy, you can look at them, you can identify differences between them, but they're quite difficult to verbalise, to come up with a verbal description of them. So we wanted to actually tap into visual memory. We didn't just want people going car, house, wheelbarrow. We wanted to see whether we could actually get them to identify the exact stimulus. So we used these novel abstract shapes. In order to manipulate participants' experience with the shapes, we first conducted a delayed match to sample task. So we had our participants perform just a standard memory test with 20 trials. So each trial began with the word remember. Then there was a memory cue, which is one of our abstract shapes, and was presented for 1 second. We have a 2-second delay, and then we have a memory probe, which is another abstract shape. So half of the trials, so 10 of the trials, were match trials in which the memory cue and the memory probe were identical. And the other half, the other 10 trials, were no match trials, in which we present a memory cue for 1 second, then there's a delay, and we present a completely novel item that participants have never seen before, a no match item. So this is where we decided we were going to be quite clever. So to go forward into the next stage of our experiment, we discarded all of the shapes from the match trials. So those are the shapes participants had seen twice, where the cue matched the probe. We discarded those altogether. The shapes that we took forward into the next phase were the shapes from the no match trials. So in this case, we have the items that were used as memory cues, which were viewed for 1 second and then actively maintained in memory. And then we have the items that were the memory probes. So these were viewed again for exactly the same time for 1 second, but were not committed to memory. So the items that were committed to memory we call the WM, the working memory set, and the items that were not committed to memory we call the primed set. Then we moved on to a repetition phase. So this is where things get a little bit tricky. We really wanted to interrogate the nature of repetition effects. So to do that, we took advantage of the phenomenon whereby neural repetition effects are attenuated as the inter-item lag increases. So if you present two items very close together, the second presentation is going to show a really, really sharp decrease in response. If you present the item, then quite a while later you present it again, you're still going to see repetition suppression, but the magnitude of that difference will be a lot less. So we see that these repetition effects decrease as the lag between presentations increases. So we used two conditions. We used an experimental condition in which we showed paired presentations of the items from our working memory set and our prime set. So these are items which were either held in memory or which were visually processed but not memorised. So in the experimental condition, these items were presented in rapid repetition. Okay, so the two items were presented 500 milliseconds apart, bang bang. Okay, so we're going to see the first presentation, and then presumably we would typically see a reduction in response to the second presentation. So that's our repetition condition, our paired repetition condition. But we needed a baseline condition. So we also included an unpaired repetition condition. There's a lot of repetitions in this sentence. We included an unpaired repetition condition in which we presented the same stimuli again twice, but with a longer lag. Okay, so these were unpaired presentations of, again, the items from the working memory set, which had been held in memory, and the items from the primed set, which had been viewed but not held in memory. So in the baseline condition, every item was still repeated twice, but it was repeated at a much longer lag. And the mean lag between stimulus repetitions in the baseline condition was about 6 seconds. Okay, so this allows us to examine the interaction between the item set, the items that have been held in memory or just primed, and the repetition condition, items that were presented in paired repetition or unpaired repetition. And what this allows us to do is to examine these two different types of effects. So if we see an increase in bold signal in response to paired repetition relative to unpaired repetition, we interpret that as repetition enhancement. And if we see unpaired repetition showing increased response relative to paired repetition, we interpret that as repetition suppression. Is everybody clear enough on that? Okay. So it's a slightly odd method to understand, but it allows us to look at this interaction between repetition and different types of memory sets. So we presented participants with these paired and unpaired repetitions in an incidental recognition paradigm. So the participants were never instructed to explicitly recognise or identify or indeed do anything with the shapes that were being presented to them. Instead, participants performed a dummy task. So we presented our paired and unpaired repetitions of the shapes, and in between, every so often we presented a fixation point. And at intervals, the fixation point changed colour, and the participants' task was just to detect the change in that colour. So the purpose in this was just for the participants to maintain their attention on the display, but they were not instructed to intentionally view the shapes or to make any kind of judgment about whether they were old or new. So just to demonstrate here again, we have the paired repetition condition and the unpaired repetition condition, and we have 4 blocks in total. We have 1 block of each of these for the working memory condition and another for the primed condition. So in the paired repetition, we can see on the top row here, we can see the same stimulus being presented twice with a 500 millisecond gap, and again down here, same stimulus twice with a 500 millisecond gap. In the unpaired repetition case, we still have pairs of stimuli being presented with a 500 millisecond gap, but it's never the same stimulus. So the gap between repetitions of the same stimulus there is much longer, on average about 6 seconds. Okay. At the end of the experiment, so we had our memory test, then we had our recognition, our repetition phase, and at the end of the experiment, we had a surprise recognition test, which we conducted outside the MRI scanner. So we presented the participants with the shapes that they had, the working memory set shapes, the primed set shapes, and some novel shapes. And we asked them to rate whether each shape was old or new, and then we asked them to rate their confidence from not at all confident to very confident. So looking at those behavioural results from that first, before we get into the fMRI data. Well, we found firstly that within the study phase, so in the memory test, accuracy was about 95%. So this tells us that the items that we assigned to the working memory set were successfully committed to memory. So we can now carry on and say, okay, these items were memorised, the other items were not explicitly memorised. We also found that target detection during the repetition phase was very accurate, over 90%. So this is participants' detection of the colour change in the fixation point. So this just shows us that attention was maintained on the visual display. Moving on to our recognition test. We assessed a sensitivity to the oldness or newness of the stimuli. This is effectively a measure of accuracy. And we found that sensitivity or accuracy and confidence ratings were both higher in the working memory condition than in the familiar condition. So participants were better at identifying items that they had memorised than at identifying items they had just seen. This is not exactly shocking. We also found that bold response during the working memory repetition blocks was significantly predicted by sensitivity in the working memory recognition trials. And this is what the figure here shows. So we have bold signal along the x-axis here, and A prime, which is sensitivity, along the y-axis. And we see that with an R-squared value of 0.268, so 27% of the variance in bold signal was explained by sensitivity in the recognition test. Okay. So moving on to the main event, really. So first we looked at main effects, and this was kind of the first thing we had to look at, but not terribly interesting. So our first thing was to look at the effects of memory condition, and we found there were no main effects of memory condition. So we didn't see that overall. We didn't see any overall increase in fold response during the working memory condition relative to the primed condition, or in the primed condition relative to the working memory condition. We did, however, see a main effect of repetition condition. So we saw increased response across both the working memory and primed items. So increased response during the unpaired relative to the paired condition. So this is effectively repetition suppression. So what we're seeing here is across both experimental conditions, we're seeing repetition suppression in a network of left lateralized regions, including the inferior frontal gyrus, anterior and posterior cingulate, you can see there, and the precuneus. Okay, so overall we tend to see repetition suppression. Again, this is not entirely surprising. We present stimuli once, we present them again. The second presentation, we tend on average to see a reduction in response. So far, so expected. Our interesting results are in the interaction. So we're looking at the interaction between memory condition, working memory and primed, and the repetition condition, so paired or unpaired repetition. So this is allowing us to look at differences in repetition enhancement and suppression across, depending on mnemonic context, so depending on whether or not the items were actively maintained in memory. And what we find is that in region of the bilateral medial temporal lobe, so including the hippocampus and the parahippocampal gyrus, we see repetition enhancement in the working memory condition and repetition suppression in the primed condition, and this is a really interesting result. So we're seeing that yes, we can distinguish repetition effects depending on a mnemonic context, on prior mnemonic experience. Okay, so to summarise experiment 1, we find firstly that repetition of items that are held in working memory results in repetition enhancement. So when we repeat those items, we get an enhancement of response. Whereas repetition of items that have just been seen but not held in memory results in repetition suppression. So we see that encoding context does influence neural repetition effects. We found that sensitivity and confidence ratings were higher in the working memory than the familiar condition, and this suggests increased memory strength for items that were held in working memory. And again, that's not surprising. If you're going to actively commit something to memory, you're going to expect higher memory strength than for something you've just viewed but haven't really intended to memorise. So our next question arose directly from this finding when we said, well, given that we see both a difference in repetition suppression effects and a difference in memory strength, could it be that this variation in neural repetition effects is directly due to differences in memory strength? There's a number of potential explanations, but we want to really test that hypothesis. So in our next experiment, we decided to manipulate memory strength and see what effect that had in our results. So we used effectively exactly the same method as before. But prior to the study phase, in which we asked participants to memorise the item for an immediate probe, prior to that we presented each of the 10 items from the working memory set and the primed set 10 times to the participants, so that before conducting the memory test they were equally familiar with both sets of items. None of the items were novel when they were first seen. Okay? So participants were familiar with all items, and the aim here was to try and equate memory strength across both conditions. So what we find here is when we conduct then exactly the same experiment again, we find that memory sensitivity, or A prime, again this is a measure of accuracy, was higher than that seen in experiment 1. So having been familiarised with all of the shapes, we find that memory performance is better. But crucially, we find that, that in the recognition test, recognition of items from both the working memory and primed set is now equal. So we have equalised memory strength. And when we then conduct exactly the same fMRI analysis following this pre-exposure of the stimuli, we find that our neural repetition effects are no longer modulated by memory condition. So we still see the expected repetition suppression across both conditions, but we no longer see this modulation by condition. So we don't see repetition enhancement in working memory condition and repetition suppression in the prime condition. So this tells us that yes, our hypothesis was supported. Mnemonic encoding experience does alter memory strength, and that's what seems to influence neural response to repetition. Okay, so those results so far are published in a paper in PLOS ONE in, I think that was 2010. Following up from that, we have experiment 3, in which we want to look at novelty and familiarity. So we have a lot of consideration here about what was it that kind of caused our original results. So we said, okay, yes, it was memory strength. The items that we have high memory strength for, the items that had previously been committed to memory, resulted in an enhancement of repetition response rather than the typical suppression of response. So we said, okay, well, one explanation of this is that repetition enhancement and repetition suppression might relate to relative stimulus salience. So our hypothesis here is that having held these items in memory in the past made these items salient. So when they were re-encountered, if you want to be kind of sort of anthropomorphise the brain about it, we're saying the brain went, aha, these are important, okay? And we see repetition enhancement in response. Novel stimuli do typically result in repetition enhancement. If you present novel stimuli and you present them even in quick succession, you're going to get a big increase in response to them. And this is because novel stimuli are highly salient. They attract additional attentional resources and they're more efficiently encoded into memory. And there are obvious evolutionary reasons why it's important to immediately recognise and attend to novel stimuli. They could be potentially dangerous or present some kind of threat. So, not surprisingly, novel stimuli tend to result in repetition enhancement and an enhanced general response to them, an enhanced attentional response. And we're also seeing now that items held in memory result in a similar response. We're saying, well, maybe this is a representation of salience. We also find that hippocampal RE, repetition enhancement, in response to stimulus novelty, decreases with increasing novelty exposure. So Murty et al. In 2013 presented participants with lots of completely novel stimuli. And originally, as they started presenting the novel stimuli, they found big enhancement effects in the participants' brains. So the participants were really attending closely to these novel stimuli. As they continued to present the novel stimuli, the participants effectively got bored with novelty. Okay, so novelty became the norm, and the individual novel items became less salient and didn't result in the same degree of enhancement. So this supports our hypothesis of salience, that we have different kinds of salience that result in repetition enhancement. So an item can be salient if it's new, and it can equally be salient if you've held it in memory and thereby sort of implied that it's important. So we wanted to test this. We wanted to compare response to repetition of two types of salient stimuli. Items that have previously been committed to memory, as in the previous experiment, or completely novel items. Okay, and we used a very similar method to the last time. So again, we used our study phase. We had our delayed match to sample task. And again, the familiar set were the memory cues from the no-match trials in that task. So exactly the same so far. The familiar set are created in exactly the same way. But rather than comparing those with the no-match probes as primed items, we're now comparing them with completely novel items. So we have our study phase, and then we have our repetition phase, which again runs exactly the same way as in the last experiment. We have paired and unpaired repetitions of familiar, previously memorised, and completely novel stimuli. And what do we find? Well, overall, we do find a main effect of stimulus set. Now, in the previous experiment, we found no overall main effect of stimulus set. We only found an interaction. Here we are finding a main effect overall. So we're finding that overall, novel stimuli result in increased bulb signal in a network of right regions, including the temporoparietal junction, parietal lobe, and so on. And we found no regions that showed increased bulb signal during familiar trials, so items that are held in memory relative to novel trials. So immediately that's very interesting. We did go on again, and we had a look at our interaction effects, looking at differences. The interaction effects, remember, allow us to look at differences in response to repetition across the different conditions. And here we're finding that in a region of the left inferior frontal gyrus here, we see increased response to paired relative to unpaired stimuli during novel trials, and the opposite pattern during familiar trials. So here we're seeing repetition enhancement during novelty and repetition suppression in response to the familiar previously memorised set. This is the opposite pattern to what we saw in the previous experiment. So in our first experiment, we saw relative enhancement of response to these items that have been held in memory when we were comparing them against items that had just been viewed but were relatively boring from a salience perspective. Whereas here we're comparing them against completely novel items, and we're finding that relative to the novel items, the familiar and previously memorised items just don't result in the same level of enhancement. We also wanted to look at the medial temporal lobe and the hippocampus, as this was a region that had shown the interaction effect in our first experiment. So we conducted a region of interest analysis to specifically just look at particularly in the hippocampus, and we found exactly the same interaction in this ROI analysis in the right hippocampus. So again, we're seeing increased response to paired relative to unpaired stimuli during novel trials, and the opposite during familiar trials. So again, repetition enhancement during the novel condition, and repetition suppression during the familiar or working memory condition. We also conducted some functional connectivity analyses. We were interested in how these variations in mnemonic context and these variations in salience would influence connectivity between memory regions in the brain. So we looked at connectivity with the left IFG region, which was the region which showed us this interaction effect between paired and unpaired repetition and the familiar and novel trials. And we found that the left IFG did show increased connectivity with the left mediotemporal lobe, including the hippocampus and parahippocampal gyrus, during familiar paired relative to novel paired trials. So what we're finding here is that enhanced cooperation between the hippocampus and prefrontal regions appears to underlie recognition of familiar items. So this functional connectivity analysis is interesting, but really it's kind of preliminary, and there'll be more work needed to be done in order to expand on that a bit further. And that paper is currently undergoing its third round of revisions at Learning and Memory, so if you'd all keep your fingers crossed for me, I'm hoping to hear good news on it soon. Okay, so just to summarise overall then, in the first experiment we saw repetition effects in the medial temporal lobe are contingent on previous encoding experience. So repetition of previously viewed items results in repetition suppression, whereas repetition of items that have previously been maintained in memory results in repetition enhancement. In experiment 2, we found that when we equalised memory strength across both of these conditions, that memory condition no longer influenced repetition effects, and this implies that the repetition effects simply reflect changes in memory strength. In experiment 3, we established that while both novel and familiar stimuli are recognised as salient signals, the inferior frontal gyrus and the medial temporal lobe act to prioritise novelty. So response to repetition of familiar items is suppressed, while response to repetition of novel items is enhanced. And we also see, crucially here, that neural responses to novelty and familiarity can be differentiated in the absence of any intentional orientation to recognise the items as old or new. So we see these repetition effects of novel and familiar items without the participants having to make any kind of judgment about the novelty or familiarity of the items. This just happens automatically without any top-down instruction. So that's it. I'd just like to particularly acknowledge my collaborator David Soto in Imperial College London and Rodri Cusick in the MRC CBU in Cambridge who provided the stimuli for the experiments, and Cleo Vidaki, who is a BSc student in Imperial who helped with the third experiment. Thank you very much. If there are any questions. Did I bore you all that much? No questions?
Ciara, you mentioned about in experiment 1 there was the working memory condition and the primed condition.
Yeah.
And it strikes me that that's a difference between explicit and implicit memory. So do you think your research has more implications for understanding explicit memory, or the fact that priming had any effects whatsoever suggests that some implicit processes were going on? So how much does implicit memory, how much does it have any theoretical significance for what you're doing?
That's actually really interesting. I haven't thought of it in those particular terms. But I think it is interesting that all of the effects that we're seeing are automatic effects. So none of the effects that we're seeing are related to any kind of top-down intention to recognise. So they're all automatic in that sense. Now, whether we could actually determine that as being implicit or not, I'd have to give a bit more thought to. But certainly they're all unintentional rather than being conscious recognition. So these effects we're observing are happening while the participants are engaged in a different task and are effectively told to ignore the shapes. So these are just automatic neural suppression effects that are kind of effectively going on in the background without any conscious recognition of the shapes whatsoever. So I don't have a direct answer regarding the implicit and explicit memory, although I think that's actually an interesting area to follow up.
Nearly all the research for almost a century, when people talked about memory, what they were really talking about was explicit memory.
Yeah, absolutely.
So when I saw your priming condition, I thought, here we go, this is an interesting, you're building in the possibility that implicit learning is happening.
Yeah, well, I'm not sure to what extent it's implicit learning, because they do explicitly look at the shapes. They just don't explicitly commit them to memory. So they're familiar because they've seen them before.
Yeah.
But they haven't consciously held them or actively maintained them in memory. So it's and I think as experiment 2 showed, it's really just a difference in memory strength. Now we could go on all day about their memory strength
So
relates
you
to.
could go on by saying, well, if somebody is shown something in an experiment, it will be unusual if they weren't questioned about it later. So so there is a possibility
Yeah.
of saying, I'm looking at this
Absolutely.
now. I know I'm not being asked to commit it to memory,
But it's probably important
but I will
anyway.
be asked about it.
Yeah, that's actually
and
a big
being
thing
asked
that we are
about it later
considering.
is, did you see this? So that's the point of getting at.
Yeah, so absolutely, and that is an issue that we've been trying to get around. So one way we tried to get around that was by having the recognition test be a surprise test.
Yeah.
So participants were told they wouldn't have to remember them later on, that this is your memory test, and then the rest of the later one was a surprise test. But nevertheless, yeah, you're in an experimental environment. It's a very different environment. We're obviously they were trying to take these very codified lab experiments and try and make some kind of application to the world. Whereas you're walking around the world, you're not going, I might be tested on that funny shape later on because I'm being shown it in a particular environment. So yes, we absolutely, that's why we're quite careful to say that there was no explicit instruction to recognise, rather than saying that the participants didn't commit them to memory. We have to say they were not instructed to,
yeah.
because we can't confirm that they didn't have a different strategy going on.
How many participants did you have?
Let me see. I think we had about 24 in the first experiment, and I think maybe 20 in the second, for the first two, and then I think maybe 20 in the last. They're a couple of years ago now, so it's, they're not big samples, but in fMRI you don't tend to get big samples for obvious reasons.
Okay, any other questions?
Sorry if I may
Yeah,
ask.
of course.
I was reminded by your study of the Sternberg. You're familiar with the Sternberg scanning one, where people were given things, a set of items to recall. You put them in working memory and they scanned them consciously.
Yeah.
And the question was whether they did it serially, one at a time, or all at once.
Oh, like a chunking kind of stuff.
Exactly.
Yeah.
So that led to a whole discussion about the evolutionary significance of working memory. But have you given any thought to the evolutionary significance of responding to novel or...
Oh, absolutely, and I mentioned that earlier on, that
Yeah.
yes, absolutely, that there's a very obvious evolutionary imperative to respond to novel items, that a novel item, any novel stimulus in the environment is potentially a threat, which is a huge evolutionary reason to attend to it. It's also potentially beneficial. I mean, you think back in the savannas or whatever, potentially something novel might be something you could eat, or something somewhere you could sleep. So absolutely, there's no question that novelty is prioritised for solid evolutionary reasons. I think from what our data is showing is that familiarity is prioritised too, because something that's familiar, and from an evolutionary perspective, familiarity is important. You need to be able to recognise, I ate those berries before, they made me sick, I'm not eating those ones again, or this person, I gave that person money last time and he didn't help, I gave him my food and he never paid me back. See, familiarity and ability to recognise is important too. But I think what our data is showing, and I mean, so it's you can only kind of infer evolutionary matters from this, it's not really intended to that, but I think we can really kind of makes a suggestion that while familiarity and the ability to recognise those things is important, that's completely outplayed by the imperative of being able to recognise something novel and attend to it. And we see that behaviourally as well, that novel items command instant attention and more efficient processing and greater commitment of attentional resources. So there's obvious reasons why that would be the case, and I think what we're showing here is that while familiarity is also important, the brain seems to act to prioritise novelty over familiarity, even when that familiarity is something that you've indicated is important by having actively committed it to memory.
Is there any, I presume you're going to use that sort of knowledge to move on, there is some sort of, sorry, someone will ask a question, with children with autism, they say that, when they recognise things, they don't have the RS response, they have more of an RE response, I think, to familiar things.
Really? I'm not familiar with that literature. I'd be interested in knowing that, but I'm not familiar with it.
Sorry, I was just wondering, did you know an application then?
No, I haven't heard of that data with the autism, although that's not to suggest it isn't there. I'm not really familiar with the autism literature, but it sounds very interesting. I have to confess that my work in this kind of area, and I've always quite enjoyed the fact that it's quite pure, that there's not, there are potential applications, but that my interest has never really been in trying to apply it. I was just interested in how the brain worked. So there are potential implications, but I'm not. I can kind of speculate here,
Sure,
but you're kind of making it up as I went along. But if you come across that autism data, if you could send it to me, I'd love to see it.
Okay. Thanks.
Okay.
Ciara, do you want to say where you're going to be taking this line of research? Does this link in with your interest in impulsivity, or are they separate lines of research?
They're separate lines of research, really. This really links in with a lot of work I've been doing on links between attention and memory. So how, for example, holding things in memory will guide attention. I've been doing a lot of work on different aspects of that. So for example, we're looking at work at the moment. So I'm doing a lot of work looking at how when you hold a particular item in memory, it'll guide attention and visual search later on. So, say for example, you're in a supermarket and you're searching for an apple. You're likely to be distracted by something that looks similar, like something that's like a lime that's round and green, but not a banana that's long and yellow. Okay, so that's items that are being actively maintained in memory will guide attention and visual search. So that's an area of research I'm interested in, and we're looking into different aspects of that. So different aspects in terms of neural response and also just behaviourally, how many items can guide attention at any one time and so on. So those are kind of converging lines of research, and I think that this method that we've been working on with this idea of the paired and unpaired repetition, and so hopefully if this paper gets accepted, this will going to be the second paper to ever use this method with the paired and unpaired repetition. So quite nice if we can kind of get that to catch on and maybe try and use that in order to investigate different effects to different types of things, like for example the effects of different manners of attention on different types of memory on attention and visual perception.
Ciara, do you have any pupillometry data on what you're saying?
I don't actually know, but I'm quite excited by the fact that you have an eye tracker here.
Yeah, because that's an obvious way of looking at attention.
Yeah, well,
during learning or priming.
Yes, exactly. And I have a master's student this year is going to be doing some work on attention work, but from kind of looking at effects of emotion on attention, and we're planning on using eye tracker to look at attention and fixation and so on, and possibly the pupillometry stuff. So, yeah, definitely.
Just think about the social psychology of attraction, and that gradually more familiar faces become more attractive.
Mm-hmm.
So when you repeatedly show photographs of random faces, the ones that people see more of, they begin to rate more attractively. It seems to run slightly counter. It would suggest that in some ways the things that we're more attracted to are things we don't actually attend to.
Well, that's one implication. I think maybe another implication, if you take that in line with other work looking at how we're tending more attracted to faces that are average and that don't deviate from the norm and so on. So the more familiar you are with the face, the more you think of it as the norm.
Yeah, well, absolutely, and there's the same evolution explanation that you feel safer with what's familiar to you, and therefore becomes more attractive. But I just thought it's like counterintuitive.
Yeah, maybe. I think all that stuff is really interesting, actually. I've never done any research on facial attractiveness stuff, although I was researching it for a lecture recently, so it's quite interesting. But yeah, I think possibly there could be actually something to think about.
Okay. I think thanks very much.